Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Inflammasomes are huge, multi-protein complexes inside cells that spark inflammation when they sense danger. Inflammasomes can also sound the alarm when no pathogens are present, contributing to diseases such as multiple sclerosis. There are many different types of inflammasomes, and the mechanisms behind how some of them form under so-called sterile conditions has been a mystery. In a new paper published December 3 in Science Immunology, Jayakrishna Ambati of the University of Virginia School of Medicine and his team show that RNA transcribed from a type of mobile genetic element called SINE causes the NLRC4 inflammasome to form when there's no infection, and for the first. This work could open up more therapeutic targets for treating dry AMD because now there are a few more players that can be targeted, says AMD researcher Yvette Wooff of the Australian National University in Canberra, who was not involved in the study. In the case of the NLRC4 inflammasome, during an infection bacterial proteins bind to a protein called NAPE, which recruits and links together several NLRC4 proteins, as well as other proteins called ASC and pro-CASPASE1. All of these components together form the NLRC4 inflammasome. Then, pro-CASPASE1 is cleaved into CASPASE1, which causes the formation and release of inflammatory IL-1 beta and IL-18 cytokines. See Pharma Looks to Inflammasome Inhibitors as All-Around Therapies, but researchers didn't know what triggers the formation of the NLRC4 inflammasome when there are no bacterial fragments to bind to NAPE. These signs are only transcribed into RNAs during times of cellular stress, such as an infection or aging. Previous research has found that when too many SINE RNAs hang around the cell, they can contribute to diseases such as Alzheimer's, systemic lupus erythematosus, AMD, multiple sclerosis, and type 2 diabetes. Researchers knew that these SINE RNAs could induce inflammasome activity, but attempts to identify cytoplasmic RNA sensors that detect SINE RNA and trigger inflammasome formation in the absence of an infection had turned up empty-handed. They transfected human cells with SINE RNAs or a control, and purified any SINE RNA complexes that formed. They identified 12 proteins using liquid chromatography-mass spectrometry, 5 of which were known to be RNA-binding proteins. They then targeted each one with siRNAs to see whether taking them out of commission would dampen the release of inflammatory signals known as cytokines, and only siRNA targeting a protein called DDX17 reduced the release of the cytokine IL-1 beta in SINE RNA transfected cells. That's the surprising thing, that there is a specific sensor for this thing. Certainly it could have been the case that there was no molecular sensor, that it was just causing structural changes or physical changes or ion flux changes that were resulting in this inflammasome being assembled, says Ambati. The researchers also determined that this type of NLRC4 inflammasome that forms under non-infectious conditions also includes NLRP3, a protein usually associated with its own eponymous inflammasome. In addition, the researchers found that when DDX17 was unavailable, there was less interaction between the NLRC4 and NLRP3 proteins in SINE RNA transfected cells. It was really a surprise that you had this double inflammasome that was being formed, says Ambati. The team also examined this system in models of disease. In white blood cells isolated from people with active lupus, the scientists found an increase in SINE RNAs, DDX17 mRNA and protein, and activated CASPASE1 compared to healthy controls. Using fluorescence in situ hybridization and proximity ligation assay, they also saw cytosolic DDX17 colocalized with the SINE RNAs in these cells. In further experiments, the researchers created a mouse model of AMD by injecting SINE RNAs under the retina, which caused retinal pigmented epithelium, RPE, degeneration, a hallmark of AMD, in wild-type mice but not in those engineered to lack NLRC4. Knocking down DDX17 blocked RPE degeneration in mice treated with SINE RNA. While he says the findings are interesting, Vavvas adds that the potential implication for diseases like macular degeneration is limited, like all studies are limited, from the imperfect nature of the animal models that we have. He points out, for example, that mice don't have maculas, the centra. He adds that, another potential conclusion of the study is the protein kinase C delta could also be a target for this type of disease. In this paper, the researchers show that protein kinase C delta is needed to help form the NLRC4 inflammasome. Apart from the findings' potential therapeutic impact, however, Ambati says, now that we know what is the sensor, at least a sensor, of SINE retrotransposons, that opens up a whole new intersection between RNA biology and immunology. Quote, "Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you."